Okay, I would like to welcome everybody. My name is Hendrik Scholl. Together with Botan Droska, I'm the director of the Institute of Molecular and Clinical Ophthalmology in Basel, and it's a pleasure to welcome you. Would like to start with a couple of housekeeping remarks. So you'll find green dots on the chairs, and everybody should sit on a chair that has such a green dot, and otherwise please change to one that has such a green dot that allows to have the social distance we are required to keep. And if we can keep the distance, you are free to put off your face mask if you want, but of course you're also free to keep it on. You can do as you wish. Sehr geehrte Mitglieder der Medizinischen Fakultät der Universität Basel, sehr geehrte Lehrende und Studierende der Universität, liebe an der Wissenschaft interessierte Zuhörer, sehr geehrte Gäste und Ehrengäste, sehr geehrter Herr Professor Rivolta, lieber Carlo. Dear members of the medical faculty of the University of Basel, dear professors and students of the university, dear colleagues interested in science, dear guests and guests of honor, dear Professor Rivolta, dear Carlo. It is a great honor for me to act on behalf of the medical faculty of the University of Basel, and as a special pleasure to introduce to you Professor Carlo Rivolta. Carlo was appointed after an international search as full professor of ophthalmic genetics at IOB. Please allow me to say a few words about him, his life and career path. Carlo was born in Monza, where the Italian Formula One Grand Prix is held, and obviously was inclined to do science from the beginning. As a child, he almost poisoned, accidentally, his dog with a solution he put together with one of these little chemist kits. Luckily, the dog survived. Later, he got interested in computers and at age 14 programmed and sold to a software house a video game simulating ski races. At the time, he earned an equivalent of 1,000 Swiss francs. To me, that proves the entrepreneurial skills and Carlo's track record in computational genetics and in silico methods, of which he is an expert of international acclaim. At Carlo's time in high school, the first serious genetic discoveries were made, to which he immediately fell in love with. Therefore, he selected the University of Pavia, since they had a program of genetics. This was the time where Ted Dreijer at Harvard Medical School made fundamental discoveries in ophthalmic genetics, which eventually was instrumental for Carlo's career. We'll get back to that. But first, he obtained his PhD in Lausanne, heavily involved in a European project on sequencing and study of an entire genome, that of the first gram-positive bacterium. We would call this genomics nowadays. While in Lausanne, he further developed his passion for computers by taking advantage of the EPFL, which eventually awarded him a degree, a sort of a master in bioinformatics. The choice to start genetic ophthalmology for a postdoc was an accident. Carlo aspired to move into medical genetics and applied to Ted Dreijer at Harvard mostly because of his great genetic papers, which accidentally dealt with ophthalmology. It could have been very well dermatology or endocrinology. He wouldn't have cared at the time. When Carlo then met Ted Dreijer in person during the pre-interview, he had a couple of other choices at Stanford and at Boston Children's. He was sure that his future would be ophthalmic genetics. Carlo arrived in Boston in the fall of 1999, just in the time to witness the collective hysteria named the Millennium Bug, the sequencing of the human genome that was completed just a few months later, the proximity with the MIT, and his background in informatics made his transition to human genomics something that was almost natural. In 2003, Carlo was appointed instructor of the Harvard Medical School and could have continued there. His move back to Europe was mostly due to the wish of getting closer to home and to grow the family's newborn kid in a less stressful environment. Carlo described to me the fact that he ended up back in Lausanne after interviews everywhere in Europe as serendipity. In late 2004, he started at the Department of Medical Genetics in Lausanne and was very happy there, working a lot, but also publishing a lot and building his international network of collaborators. Carlo indeed has access to large patient cohorts in the US, Europe, South America, and East Asia. It was in 2017 when he was appointed as Professor of Medical Genetics in Leicester, United Kingdom, at the mythical Department of Genetics that saw the discovery of variable DNA repeats by Sir Alec Jeffreys, starting several fields of investigations from forensic genetics to population genetics, etc. He was about to go there, but then, very luckily for Basel, he was headhunted by me as an outstanding candidate for the new position at IOB, and everything fell into place for him and for IOB. Carlo's arrival at IOB in mid-2019 marks a cornerstone in IOB's still young history. He has established a research group that pursues an ambitious interdisciplinary research program. The group focuses on discovering the genetic cause of monogenic and genetically complex ophthalmic diseases, and the research team is particularly interested in defining the mutational landscape of retinal ciliopathies, which is a part of the retinal photoreceptors, as the presumably most prevalent cause for hereditary blindness in Switzerland and in general in Europe. Their research includes exploration of modifier genes and their effects on therapies, the development of mathematical theories of visual function, the creation of computational tools for elucidating the genetic causes of visual loss. The Ophthalmic Genetics Group comprises considerable expertise in genetic investigation, particularly in high-throughput techniques such as parallel genotyping and next-generation sequencing, as well as in disease gene identification, computational and in silico methods. Carlo has managed an extraordinary growth of his research group, which, after less than a year, includes more than a dozen people. Carlo has appointed 6 PhD students alone just in the past few months. Since the beginning of 2019, Carlo has authored 23 peer-reviewed scientific articles, including, amongst others, 3 times Human Molecular Genetics, 2 times Nature Communications, and also PNAS, amongst others. But Carlo's importance for IOB goes even beyond. As an example, Carlo was instrumental for the acquisition of IOB's espresso machine on the 7th floor of the lab building at Klebeck. I can only recommend those who appreciate real Italian espresso to stop by and enjoy a cup of coffee together with a wonderful view over Basel. In these unprecedented times, it is impossible not to refer to the COVID-19 crisis. All the more, I am happy to say that Professor Rivolta is not only at the forefront of gene identification for ophthalmic disease, but also in COVID-19 research. He is co-principal investigator of a research project funded amongst the first of the Swiss National Science Foundation, together with Professor Pierre-Yves Bauchu, head of the infectious diseases immunogenetics team at Lausanne University Hospital. The project is entitled Host Determinants of Severe COVID-19, Learning from Extreme Cases. The hypothesis for this project is that severe forms of illness after SARS-CoV-2 infection result from individual genetic predisposition of patients. The researchers will therefore study the influence of genetic factors on the occurrence of complications of COVID-19, especially in young subjects. This could allow specific or even prophylactic treatments for patients with higher risk of severe development of disease. More specifically, DNA samples from these individuals will be collected at the CHUV and other Swiss hospitals, including Basel University Hospital, and Mathieu Quinaudot, postdoc in Carlo's team at IOB, will analyze their genomes to search for common or rare DNA variants that could make them prone to develop severe symptoms. Ladies and gentlemen, please join me in welcoming Professor Rivolta and to attend his lecture entitled Ophthalmic Genetics, Keep Your Eyes on the Computer Screen.
Lieber Professor Scholl, vielen Dank für Ihre lieben Worte. Liebe Kollegen, Freunde und Familie, liebe Gäste, vielen Dank, dass Sie heute persönlich hier sind, trotz aller Umstände, die mit dieser Pandemie verbunden sind. Ich bin dem IOB und der Universität Basel sehr dankbar für die Ehre, heute hier sprechen zu können. Leider endet meine Rede auf Deutsch hier auch schon. Ich entschuldige mich aufrichtig dafür und hoffe, dass dies für niemanden ein Problem ist. In fact, my German is beyond bad, is basically non-existent. And I had to read this, of course. Thanks very much again to all of you for being here. I am very pleased to be here. I'm actually honored to be here. It's a privilege to talk in front of you about what we do in the lab. And I am particularly grateful to Professor Scholl, the directors of IOB, for giving me the opportunity, and of course, University of Basel, for giving me the opportunity of developing my research in this beautiful place, which is IOB and Basel. So, I remember that once upon a time I read in a book, which was a novel, not a scientific book, that every scientific lecture, every scientific communication is usually divided into 3 parts of equal length. The first part can be understood by everybody in the audience. The second third can be understood by colleagues and people from the field. And the third part can be understood only by the one who speaks, meaning me. So I tried to deviate a little bit from this scheme, and I prepared something which is probably 80% understandable by everybody and 20% to my colleagues, because after all, I have to show some scientific data, not just general concepts. But I'd like to start from something very basic. So, as you all know, our body is made of a cell, and every cell has a nucleus, which is this orange circle here. And in the nucleus, you can find chromosomes, which are nothing less than very highly compact structures of DNA and protein. And if you disentangle the chromosome, if you roll them out, you get to see what is the universally known double helix, that in fact is made by 2 helices and also contains the basic letters of the alphabet that composes life, A, C, T, G. And again, as most of you know, if not all of you know, these letters contain information. They can be read as words, as sentences, or as pages by the cell. So if I dare to make a parallel between the DNA and basic biological processes with something which is more common to everyday life, one can say that the DNA is like a cookbook. And in fact, the whole content of a cell nucleus, the genome, is the whole cookbook. In fact, we have 2 cookbooks, one coming from our mother and one coming from our father, which are slightly different. But in the end, is more or less the same thing. Now, in addition to the cookbook, we have something which I call the Unimaginative Chef. So in order to make sense of the cookbook, you need somebody to read it, understand it, and apply the instructions that are in the cookbook. And at the molecular level, this is some very complex macromolecular machinery that I'm not going to talk about. At the more common level is really a chef. The key word is unimaginative. So this chef can read the instruction but does not interpret them. If the cookbook says cook for 10 minutes, it's going to be 10 minutes, 0 seconds. He or she doesn't have any imagination, just applies to the letter of what is written in the cookbook. And of course, the end point of the process is a chocolate roll, a meal, a cake, something that you like and that you want to eat. At the molecular level, this is represented by proteins. Nobody cares about the cookbook, nobody cares about the chef, some maybe, but everybody cares about the chocolate roll. The chocolate roll is what you want to have, what you want to eat, what you care about. And for the cell, this is exactly the same. What is important for the cell is not really the cookbook, but the protein. Proteins are things that make us. All visible characters from us are determined by the activity or the presence of protein. And I will get to this in a second. Now, if we take the image from before, we can imagine that sometimes you have slight changes of the DNA sequence, which we call mutations, to be as vague as possible. These are small changes in the sequence of the letters, and this can occur every day, every minute. It's pretty frequent to have mutations in our DNA. And what happens is that a letter is changed to another letter by, for instance, here I indicated UV beam of light from the sun on your skin or something like this. But it can be really anything. It can be whether you smoke, whether you eat something which is not good for you, or simply oxidation that is currently occurring in every cell. And if we go back to the parallel, we can see that a mutation here can be assimilated to a typo in the cookbook. So, for instance, instead of saying cook for 10 minutes, the mutations can say cook for 90 minutes. So 1 becomes a 9. And the unimaginative chef doesn't make anything more than reading the instructions and really cooks for 90 minutes and 0 seconds. And the result is that you have some bad chocolate roll, maybe eatable, but not as appealing as the previous one. And at the molecular level, this translates into a protein. It doesn't do its function. Again, we usually do not care about the cookbook. We really care about the cake because, in fact, we are the cake. The fact that we may be, for instance, intolerant to lactose, the fact that we have specific sensitivity to some medications, the fact that we have blue eyes, all these things, all these characters are determined by proteins, not by the DNA. The DNA is just instruction for making the proteins. And having this in mind, it's not difficult to think that for many, many centuries, many, many years, all medicine and biology also has cared about proteins, about visible things, about things that you can intervene on very, very easily. For instance, if we take the example of this ear, here indicated with this pretzel, and if the pretzel is not really perfect, then a physician can say, okay, so maybe I can do a little bit of surgery and I can correct this ear by modifying a little bit the shape of the pretzel. Even a biologist would be more interested in, for instance, in understanding why this pretzel is not correct. And perhaps, I don't know, count the grains of salt on the surface of the pretzel and, giving you an elaborate theory by which you have 45 grains instead of 50 or something like this. Conversely, geneticists care about the cookbook. they're more interested in the instructions than in the final product. And, geneticists are a bit bizarre with respect to the rest of the scientific community. And ophthalmic genetics, the chair I had the privilege to get here at the University of Basel, is a subset of medical genetics, which itself is a subset of genetics. So genetics is the science that studies everything which is determined by DNA, for instance, the color of Mendel's peas and things like the shape of leaves of plants and stuff like this. Medical genetics is interested in studying exactly the same things, but for characters that have to deal with diseases, with hereditary conditions of any organ or tissue. Can be the skeleton, metabolic diseases and things like this. And ophthalmic genetics, as Henrik was alluding to before, is just a subset of medical genetics that care about mutation that lead to dysfunction of the eye. So here is coming a little bit of a technical slide showing a little bit some sort of formalism that I have to introduce for you to understand the rest of the talk, at least some of it. If we get back to the example of the G mutated into an A that I was showing before, we can see, for instance, that the presence of the mutation, the A, determines something in this individual who has the A in his genome. So this is a character. For instance, let's say it's a genetic disease. If you see somebody who is red, it means that he or she has a genetic disease. And you can see that somebody who has the disease is because has, at the genome level, the A mutation. Conversely, those who don't have any mutation are healthy and are indicated with a blue color. So if we do this in sort of a medically more formal scheme, you see that this is exactly the same. So men are squares, women are circles, and it's exactly the same scheme. And whenever you see a black symbol, it means that this person has a visible character, in this case, a genetic condition. So what we do, we study inherited retinal degeneration, IRDs in short, that are hereditary diseases that, as you will see in a second, can lead to blindness. And that touches specifically the retina, which is this yellow layer on the back of the eye that contains the cells that understand who collect the light signal from the environment and make the image that we perceived to be sent to the brain. These diseases cannot be corrected by optical system. For instance, you cannot improve it by having glasses because the retina is, you can imagine it as a screen of a computer. You can have the best optical system, but if the screen doesn't work, well, the image is distorted and it doesn't really help you having better glasses, things like this. So what is it really? Here I'm showing in an extremely short period of time some signs and symptoms of patients with RP or retinitis pigmentosa, which is the most common form of retinal degeneration. Patients with RP first, typically, experience problems in seeing at night. So they have night blindness, which is simply, what is dark is darker for them, and what is bright is sometimes too bright, kind of painful to be seen. And as the disease progresses, also daytime vision is lost progressively. This is a progressive neural degeneration. Every year is worse than the previous one. Actually, every hour, in theory, is worse than the previous one. And in daytime conditions, they tend to lose peripheral vision to be very, to summarize to the maximum, and to what is called commonly tunnel vision. So you have a tunnel, and you see only in the center. And as the disease progresses, this area of vision gets smaller and smaller to the point that maybe in some patients this tunnel closes completely and the person becomes blind. And from the physician point of view, This is a back of the eye as seen by an ophthalmologist who looks into somebody's eyes. And although I'm not going to go into this, because there are better people than me who will be able to do this, you can appreciate that also at the macroscopic level, this fundus, which is from a control, is not the same of this fundus that is from a patient. So major changes induced, again, by genetics, and genetics is exactly this. So we can have these diseases as the results of the presence of a single mutation, indicated here with an A, exactly in the example that I was showing before. This is called dominance. So if you have one mutation, automatically you get the disease. It can be recessive, meaning that you have to have two mutations in the same gene, so two typos in the cookbook from your mother, one from your mother, one from your father, in the same, let's say, recipe, which is the gene. So the recipe of the cake, of the chocolate roll, has to have typos in the maternal cookbook and in the paternal cookbook in order for somebody to develop the disease. And again, you have two mutations, one from dad, one from mom, and when they are together, the disease manifests. If you are carrying one of the two, nothing happens. And finally, there is a minority of cases which is due to mutation on the X chromosome, which is present in two copies in women and in one copy only in men. And the mechanism is more or less the same, meaning that in girls, the presence of two X chromosomes, if you have a mutation in one of the two cookbooks, then the other cookbook can compensate for the presence of a mutation in the first one. Conversely, if you are a boy, well, you have only one. So if you have a mutation, then obviously you get the disease. The classical example of this, which is definitely by far not a condition, is color blindness, Daltonism. Only boys usually have Daltonism, not girls, apart from exception. Another typical feature of IRDs, of retinal degenerations, is that they are extremely rare. We did recently a calculation based on population genetic burden, and we ended up with this figure, which I tried to represent in this donut chart. 1 in 1,600 individuals has this disease, which is considered to be rare. Luckily for all of us, this is a rare condition. And finally, probably the most interesting characteristics of this is that in contrast to most genetic conditions that are rare, IRDs can be caused by mutations, by typos, in more than one recipe. You can have the chocolate roll that is defective, you may have the apple pie, you may have the spaghetti, whatever you want. So it doesn't matter what meal is affected by these typos, as long as these meals are comprised into this spectrum of, recipes, let's say. So an important consequence of this concept is that if you take cystic fibrosis, which is the classical monogenic rare condition that everybody knows about, cystic fibrosis is caused by mutation in the CFTR gene, only one gene, not as many as this one. So only, let's say, the recipe of the chocolate roll. So if you observe a person who has a disease, you immediately know that this person has a mutation in the CFTR gene because you don't have any choice. It's this gene, period. Conversely, so if somebody is healthy, then you know that there is no mutation in this gene. And the opposite is true. If by some strange reason you could look only at the genome but not at the person, you would know that if you find a mutation in the CFTR gene, then the genome is from a person who has cystic fibrosis, and if you don't find any mutation, then the genome is under control. For IRDs, things don't work this way, unfortunately, or maybe fortunately for our research. If you have a person who has no symptoms, very likely he or she has no mutations in the list of genes that I showed you before. However, if you have a patient, if you see a patient in the clinic, then you don't know exactly what gene is mutated. You know that one of them must be mutated, but you don't know exactly which one. And if you see two patients, then things get even more complicated. So unless they are from the same family, you don't know whether they have the same mutation in the same gene, different mutation in the same gene, different mutation in different genes. There's no way of knowing it unless you really go and look into the cookbook. So what do we do to tackle this problem? In a very short and summary slide, we collect blood and biological samples from patients with IRDs from around the world, including Basel and Switzerland. And we analyze this genome by means of computer analysis, and I will be a bit more specific in a few minutes. We do some genetic testing, and sometimes we do functionally validate this mutation, meaning we do try to understand why the typo in the cookbook results in a failed cake. And this leads to the identification of novel disease genes, of the understanding possibly of new inheritance patterns, and also we are trying, since recently, to develop new tools for data analysis, so for finding these typos in the cookbook. But at one point, we will have to read the cookbook, because this is a condition you cannot escape. And how do we do it? So up to a few years ago, there was a very, very complicated thing to do. The Sanger sequencing system, which you see here, was a wonderful system to get the sequence of the DNA, but it was extremely slow and extremely low throughput. So in the example of the cookbook, it was, as you could read, for instance, one line of the book at the time maximum. And by the way, this is Fred Sanger, Nobel Prize for Chemistry, who invented that technology. Nowadays, we have the privilege of using what is called next generation sequencing, which is a complete revolution, a technological revolution that is started commercially about 10 years ago, that allows us, without entering into details, to sequence every single base of the genome, meaning reading every single letter of the cookbook, of the two cookbooks. The problem, though, so you may say, okay, so we have the cookbook, we can extract the DNA from a person, we have means of reading it, so everything should be easy. Well, it's not that easy. And I'm going to illustrate this by a couple of examples. And if you have to remember one slide of this talk, please remember this one. The human genome is very, very big. So these are two standard trash bins from city of Lausanne. And the standard, not in Basel you don't have these things, but in Lausanne you do. And imagine then to be full, as my two daughters are illustrating, by grains of sand, completely full with sand, like beach sand. Now, a single grain of sand would be a base of the DNA, and this would be the genome. So our task is to find one grain of sand, the mutation, in here, and only one. We don't care about the billions that compose the rest of the genome. We just have to be very precise and identify this specific grain of sand. Seems not that easy anymore. In addition, the genome is very, very variable. We are all different. We have variants in the sequence of the DNA that are not harmful and that make what we are, but still they are poisoning the system because we don't know whether these variants are actually mutations, benign variants, things that have nothing to do with the disease we are studying. So not only is big, but it's also very variable. So we can say that genetics is like puzzles very much, because the needle in a haystack is not really, I mean, it's a good parallel for this specific kind of analysis. And if we do whole genome sequencing that I was mentioning before, so if we sequence the full genome of a person, we end up with 4 million grains of sands that are bizarre. They are not really match the human genome reference sequence. And if we are trying to find a mutation, one mutation in a patient of whom we have sequenced the full genome, well, we have one mutation and 3,999,999 false positive. So even if we can reduce the number of positives to 4 million, it's still a lot of work to do. Luckily, we have means of doing this as well, which is represented by computers. So my lab, as Hendrik was alluding before, is slowly but steadily transitioning from the classical wet lab to something that looks more like this, because the genome is complex and the genome is big. And the key element, hence the title of this talk, of the process is this guy, is the computer. Yes, we have the patients. Yes, we can get the blood and the DNA. Yes, we can sequence the DNA. But the bulk of the work is done by the computer. And here I'm just showing a very simplified and naive flow chart that represents what we do every single day by what we look at the genomes. So again, not that easy. And the process looks more or less like this. So if we take the variants that are present only in the coding part of the genome, so let's say meaningful parts of the cookbook, which is less than 4 million, but it's 100,000, we can think about ways, or the computer can operate some filtering to say that, okay, so if this is a mutation, it must have an impact on the protein because I want a fail cake. Therefore, this reduces the number by 1/6, more or less. It has to be rare because these are rare conditions, so we don't care about common variants because otherwise we would have a very high number of patients, which we don't because we don't observe them. And again, we reduce even further. Then we can apply some quality control criteria and so on and so forth. So we end up with something which is manageable, like 3 to 10, 20 variants per person in the case of a recessive condition. So that was the 80% of the talk, and I hope that you enjoyed it. Now we are getting to the 20% more technical, because again, I have to at least show you some data, I guess. But I'm going to do it in along the same lines of the introduction that I showed you. So the first example is an example of 2 patients, 1 and 2, one from Greece and one from Sweden. And not sure whether the resolution of this image is good enough, but they do have retinal problems that touch the central part of vision rather than the periphery. And they also, as you can, well, probably you cannot see, but is shown in this by these 2 graphs, they have slight hearing problems. Not so the gray area is how a normal control person could hear, and these other red and black lines and the way they hear. So it's slightly worse than most of us. And fine, we thought that this clinical presentation could be interesting enough, and then indeed we sequenced them. Again, 2 different countries, definitely not related. And we went through the same process that I showed you before. So we sequenced all the exonic variants, then we filtered by those that had an impact on the protein. We filtered for rarity, and so on and so forth. And at the end, we got only 2 genes that had mutation-like DNA variants in the genome of these 2 people who were so far away from each other. Now, TTN is a gene that is mutated, that has variants in essentially every one of us, so we knew that it was a false positive. Sub78, we kept it because it was something very interesting. And then we went back to the families, again, these schemes, and we looked at the presence of mutations in this gene indicated here by M1, M2, and M3. And you see that every time you have a person with a disease, this person carries this mutation, 2 copies, maternal and paternally. So we are pretty confident that the things were working correctly. And then we went functional, and we look at the retina. We could stain, stain is understandable, the protein that is made by this gene, indicated here by these red dots. And we could for sure see that this protein was localizing at the cilium, the connecting cilium of photoreceptors, and in particular at the connecting cilium of cones, which are specialized photoreceptors, which are sensitive to high intensity light and allows us to see in colors. And the green color, basically, it's a marker for cones, so we were pretty sure. And since this was a ciliary protein, we actually went further and took some skin from these people and look how their fibroblasts, so this is a control and these are the 2 patients, how their fibroblasts cope with this mutation, how their cilium is presenting. And we saw that their cilia were actually longer than the cilia of controls. And in the ciliary world, having a longer cilium is not good. It's actually a sign of something which is not functioning. Second example is a more recent one, which is again following the same structure. So in this case was a syndromic presentation of patients from Portugal and from Brazil. In this case, we had 4 individuals who had the disease, and the clinical presentation was pretty particular. So there were some short stadia of kyphoscoliosis, some intellectual disability, some hearing loss, and some radiological signs, but of course, visual impairment, retinal degeneration, and in this case was Leber congenital amaurosis, which is a very severe form of retinal degeneration. And then somebody very smart, not me, realized that the same presentation, the same signs were described in a case report of a little lady from the Azores Islands, who in 1986, so a while ago. And what was shown was pretty reminiscent of what discovered recently, and including the radiological signs. And again, we did the same thing. So we took the 4 patients, we look at regions that may have commonalities, and we identify some teeny tiny region on chromosome 22. We looked was present in this region. We, of course, did genome, in this case, exome sequencing, and we realized that all of them had mutation in a gene which is called PISD. And in this case, the mutation was not a change of a base from G to A, but it was a deletion, meaning that some piece of DNA material was actually taken off the genome, as you can see here. So this is the control sequence of the DNA, and you see that patients had a piece of DNA completely missing. And of course, it did, we call it co-segregate, so the presence of mutation was the same in all the patients, but not of those who were healthy. And we were also lucky enough to get back to paraffin-embedded sample of the original patient described in 1986, who unfortunately died in the meantime, but we could get DNA from the paraffin, and this DNA was also positive exactly from the same mutation. So we could confirm that these 3 different families from 3 different geographical areas had indeed the same mutation in a model that could in fact be made. And you can think that, for instance, the mutation occurred in Portugal a few hundred years ago. This person then, had offspring in Portugal, and then we could actually find the descendants of this original person. Maybe some of them migrated to Azores in their way to Brazil, when the Brazil was colonized by Portuguese, and this is why we could actually see the presence of the mutation in these 3 different geographical regions. But getting back to the point, so why genetics is important? Well, it's important in general, I guess, but it is specifically important now for ophthalmic conditions, because since a few years, we have the possibility of intervening in the cookbook. I said before that usually you have this unimaginative chef, and then the chef is imaginative. The chef remains unimaginative, but since a little more than, since a few years, let's say, we can go and edit the cookbook. We can correct the cookbook, and this is done, for instance, in the case of retinal condition by injecting viral vector, which is a little machine that puts the good copy of the DNA into the genome by injecting it directly on site or intravitreally. Well, anyway, the delivery is not that important. So in principle, we could treat people with defects in the cookbook. Problem is the same. Which gene are we going to modify? We don't know a priori if a person has a defect in gene A, B, C, D, E, Z. And again, therefore, we have to start from the genome. We have to look at the grains of sand, and we do this by using heavily our wonderful computers. We, if you are lucky, say, of course, it's gene D, and then we can finally intervene on the patients. And this is extremely important because we are working at IOB. And IOB researchers and clinicians united to restore vision is exactly made for this. So geneticists at IUB are part of a process that has as a final goal to treat patients and give them vision back, hopefully. So yes, the patient is seen by the ophthalmologist at IUB. By the way, the ophthalmologists think they are the absolutely most important people, because without them, patient wouldn't be diagnosed. And then the information is given to the geneticists, and they think they're the most important people of all, because without them, even if you have the patient, then you would have no information. And then they give the information to the bioinformatician, who say, okay, fine, you have the DNA, you have your genetics, but if I cannot run the computer, you will never find the gene or the mutation. Therefore, we are the most important people in the process. And finally, this information is passed on to the biologist, which is, of course, the most important person, because he or she is the one that devises systems to get back to the patient. So it is really a system when everybody is extremely important and everybody plays a fundamental role for, again, helping patients with their problems. So, in conclusion, full genomic sequencing allows us to read the cook, but this wealth of information is paradoxically a problem because we have too much information to deal with. Luckily, there are computers that help us in this process, and one corollary thing that I can say is that the era of non-computational genetics is probably gone forever, because the geneticist of 2020 has to be able to deal with computer science. And finally, ophthalmic genetics is an essential, although maybe less spectacular than growing cells and making injections in the eye. That is nonetheless essential for understanding the mechanism, but also for guiding the process of intervention into patients. And finally, last but not least, I'd like to acknowledge a lot of people. I'd like to acknowledge my past lab, the current lab. To paraphrase placebo, scientifically speaking, without you, I am nothing. Really. And IOB, of course, because it's a wonderful environment. I'm super happy to be able to work in this environment. And close family and extended family, thanks very much to you as well for enduring my weekly commuting to Basel. And thanks very much to your attention. Thanks.
Carlo, thank you very much for this wonderful lecture. I hope you learned a lot. At least I have learned a lot, and actually maybe I shouldn't, because I should know all that, but it speaks for the quality of the lecture that even I, being so familiar with Carlo's work, learned quite a bit. Because I represent IOB and only today on behalf of the medical faculty act here to introduce Carlo, I would like to deviate from the protocol that is given to me, because now would be the time to hand over flowers. But yeah, we have a specific opportunity today. Because Carlo invited close and extended family, we have the opportunity to hand over flowers to his wife, to Carlo's wife, Emma, and I would like to deviate from the protocol and hand them over. Kisses not allowed. Last but not least, I would like to thank you again for coming to Carlo's introduction lecture. It will not be the last time that you've seen him, given his past performance over the last year only. We will see a lot of him in the future here in Basel. Today, we from IOB would like to invite you for the apéro that will take place in the entrance hall of the museum. We hope you like it, and again, congratulations, Carlo, to become professor at our faculty.